Hello everybody and welcome to today's webinar, Risk-Based Assay Development Strategies for Cell Therapies, hosted in partnership with QualTex Laboratories and BioBridge Global. I'm Georgie Makin, the editor at Facilitate and moderator for today's discussion. Today I'm joined by a great panel who will shortly be introduced to you, but before we get started, I just wanted to take a second to remind you that the latter half of this session is reserved for an interactive Q&A discussion, so please do submit your questions via the Q&A widget on your control panel. So without further ado, I would like to hand over to Ward Carter to start today's session.
Thank you, Georgie. I appreciate it. Hello, everybody. Welcome to our webinar. Thanks to Facilitate for giving us an opportunity to present this information to the audience today. We're really looking forward to an interactive and hopefully beneficial dialogue with everybody that's chosen to participate today. We really do hope that by going through the risk-based assay development strategies for cell therapies, that there will be some information here that will be very helpful for any of the participants that may be looking at or are in the process of developing a cell therapy. So to introduce the panelists today, my name is Ward Carter. I'm the Chief Operating Officer for QualTex Laboratories and Senior Vice President for BioBridge Global. We have Scott Jones, Vice President of Scientific Affairs, that will be presenting today, and Emmanuel Casasola, who is our Executive Director for Global Quality and Compliance for BioBridge. So without further ado, I will turn over the discussion to Emmanuel.
Thanks, Ward. Appreciate it and hope everybody's doing well today. So when therapeutic developers start down their journey, they typically first think about their collaboration partners in terms of manufacturing and regulatory and clinical trial support, and with good reason. But sometimes what is overlooked is getting a good testing services partner. A good testing services partner will allow the facilitation of the testing that's necessary to either support the clinical trial results or prove efficacy of the therapy. So you really want to try to partner with a good testing partner as early as you can in the development of your therapy. Now, when we engage with therapeutic developers, there's some common risks that a lot of them face, and the ones that clearly stand out to me are number 2, selecting a lab that doesn't understand the assay life cycle. Number 4, appropriately underestimating the lead time and lab capacity that impacts the development timeline. And then number 7, the not considering all geographies targeted for commercial application of the development of the therapy. So the assay life cycle is the development, the qualification, the transfer, and the optimization of a test. Now, the life cycle is critical in ensuring that the testing will withstand the scrutiny of the regulatory process. So it's really, it's necessary for a laboratory to be able to guide the discussion with you so that you can properly understand the phase appropriateness for the testing life cycle. Unaccounted lead time can also cause delays, which can both be compounding and can be, critical with the financial aspect and also the lead time to getting something to a clinical trial or commercialization. Now, some of those accruals might be from various variables, such as obtaining materials, the laboratory capacity itself, the manufacturing process development, any tech transfer that might be necessary, and also quality system integration. So you really need to make sure that you estimate that constraint accurately. Lastly, you should consider the regulatory requirements for the current stage of the therapy and the area for where the therapy is going to be commercialized, and all those different aspects in between. Next slide, Ward. So even though there are active efforts to harmonize the industry, especially through ICH, the International Committee for Harmonization, there's still differences that exist between various regulatory geographic regions. Therapeutic developers are ultimately responsible for adhering to the regulatory requirements for the therapies. However, you can mitigate any risks by working with partners who actually understand those nuances or can walk you through those differences. But one would ask, what could be different? Well, there are differences in IDM testing, which stands for infectious disease markers, and that's specifically on the source material for where your cells are going to be derived from. There may be unique requirements regarding acceptable facilities for aseptic processes or sterile processes. There are varying expectations for data integrity as well, so you'll want to make sure that you understand those accurately. One of the biggest differences, though, is going to be on the expectation for potency assays. The onus is on the therapeutic developer to understand what those are completely. However, once the regulatory body provides those expectations, or once those expectations are understood by either you or your partners, the right laboratory partner can help you qualify what that assay is, how it works, what are the differences between the results that you're getting. It's not a good idea to assume that you know, especially for potency assays. So you want to make sure that you understand those completely. Now, there are going to be some similarities, and some of those assumed similarities are going to be things like safety and purity assays, because there are pharmacopoeia standards that most developers can follow and go down the regulatory pathway, similarly. I put in some examples of just from a facility standpoint, the differences that you might encounter, and this is for clean room classifications. Now, these come straight from guidance documents from the FDA and from the EU, but you can see that in a grade B environment for the EU, you have some overlaps between ISO grades 5 through 7. And so just understanding the manufacturing aspect and then the testing parameters, all of those it might be unique nuances, so you want to make sure that you understand those completely as you go down the road. All right, go ahead, Ward. So when we engage with therapeutic developers, we start off by determining what assays may be available for testing of their products. We want to understand the analytical target profile. You may also hear that as ATP. So some of that is just understanding what assays we have available for testing, and then determining what do we need to do in order for the assay, for us to be able to test that assay. And this is just a sampling of questions that we may ask, but some of these are the things that, as a therapeutic developer, you want to take into consideration. So, is the assay being offered? Is the intended use of that assay different than what it's going to be doing ultimately to prove for your product? Are there any new or different materials that are required for the assay? And this may be in anything in pre-analytical or in the analytical portion of the testing. Are there any different environmental conditions needed for the assay? Does it need to be in a CO2 type of environment? Does it need to be in a sterile environment? So there are different environmental conditions that need to be adhered to depending on what the test is for. What's the pre-analytical process? What are we having to do to prepare the cells or prepare the product for testing? Do we have to expand? Do we have to do different things from a pre-analytical standpoint for us to be able to test it? What's the instrument configuration? If it's on a flow or if it's on some kind of PCR type of assay, what are those instrument configurations needed to be? And those need to be changed in order for us to test accurately. And then from a data integrity standpoint, what's the software configuration going to be? How can we ensure consistent results for the interpretation of those results so that they can support clinical trials or the potency assay portion of it that's proving the therapeutic? And then, are laboratory personnel proficient in the analysis to obtain reliable results? Is that repeatable? Can the testing be conducted by people who are doing this day in and day out, and do they have proficiency in that? So like I said, this isn't all inclusive, it's just a sampling, but some of the things that you want to take into consideration when you're going down the road of what kind of testing do we need to have, and what kind of partner are we needing to ensure that we can get reliable results for our therapy. So now I'm going to turn it over to Dr. Scott Jones for custom assay development.
Thanks, Emmanuel. Let me go ahead and I'm going to talk some more about a lot of specifics on custom assay development. And really kind of the first question is, why is assay development important in the first place? Testing of cell and cell-based products to demonstrate safety, purity, and potency is required by regulatory authorities. So obviously very important. Usually pre-existing assays can be utilized for some of the testing, but there's always some degree of custom assay development is usually required, especially regarding potency testing, and we'll talk more about potency testing in a minute. And in recent years, the cell and cell-based therapy industry has experienced increasing capacity constraints in routine product testing and analytical assay development. And these constraints have caused several issues, specifically delays in product development, product supply, clinical trials, regulatory filings, and approvals. So obviously very serious issues. So really what I kind of want to focus on is, in kind of the whole meeting of this, assay is, or excuse me, this talk is really to talk about some of the testing strategies that can be used to mitigate this risk. Obviously, first of all, talk a little bit about the assay development process and having a robust system and how important that is. Focus on potency assays. You've already heard Emmanuel talk about them. I will talk about them again. So kind of our mantra is potency, potency, potency. So we'll talk about that. And then also kind of looking at different approaches to, especially to safety assays. And that is the idea of using qualitative assays versus quantitative assays, and also sort of using automated infectious disease monitoring assays that specifically have been used in the blood screening world to make life a little bit easier, and we'll talk about that too. So now I'm going to go over each of these strategies in more detail. And so let me go ahead and go to the next slide and then start talking about assay development process. Or if you can go ahead and, thank you. So as you can tell by this list, there's really 6 kind of key steps in the development process. They're outlined on the slide. And really you're starting with the original specifications going all the way to what is also important, and that is, management of the system once you have it up and making improvements to it. Obviously, there's a lot of things to consider. If we can go back one more. There's a lot to consider in here, and so we do list some of the sort of key concepts under each of those steps. Obviously not going to go into all the detail on them, and also, this is just some of them. This is obviously not all of the things that are required as part of this process. And I know this is a busy slide, but really there's kind of 4 key things I want to make sure that I convey today. And that is, as you can tell by this list, there's a lot to consider when developing and implementing assays. It's not just a few things, it's actually a relatively complicated process. You really need an experienced lab partner to do this. Obviously, having experience and understanding these and knowing these type of things is very critical to getting your therapy or product out in a timely manner. And as you can sort of guess, this process takes time. It is complicated. So the bottom line is, the earlier you start, the better. Just like Emmanuel showed in the little pyramid diagram, it's just as important to start talking to your testing group as it is to regulatory and also thinking about manufacturing. And kind of the last point is, whether designing assays from scratch or you're doing tech transfer, meaning the therapeutic developer has assays that they're moving to the testing facility, the bottom line is the laboratory needs to make sure that the assays are sustainable, scalable, and repeatable. And so a lot of times, especially if you're transferring assays, there are adjustments that need to be made for this. So once again, it gets back to the concept that it takes time, and obviously the earlier you do it is better. So I can't reiterate that enough. Let me go ahead and go to the next slide. And what we're going to talk about here is talk about criticality, or talk about potency assays. So, what is potency? If you look at the FDA's definition, which is listed here, it's really the specific ability or capability as indicated by appropriate laboratory tests or by adequately controlled clinical data obtained through the administration of the product in the manner intended to affect a given result. So as usual, not it's clear, but a little bit hard to understand. So we kind of like to think about it as potency really is the ability of a product, in the case of a therapy, to do what it is supposed to do. And so what's interesting is, based on this definition, potency really is unique in being the only critical quality attribute, or CQA, that's linked directly to efficacy of the product. So that's really why FDA and other regulatory groups put such an emphasis on potency assays, is this is the one true link that is showing the efficacy of your product. So the bottom line is, you do have to have assays to prove the potency of your product, and this proof is needed for regulatory approval. So without validated potency assays, a therapeutic product cannot be licensed. And this, as demonstrated by the FDA's recent position regarding a cell therapy showing great clinical benefit, but lacks sufficient proof of potency, and was not, given licensure. So very critical, and that's why, anytime you hear a talk about assay development in this industry, you're going to hear discussion over potency. So let's go ahead and sort of show an example of potency. So if you can go ahead and go to the next slide. So let me sort of set the stage. So here's, go over sort of an idea of potency. This slide really shows the characterization assay. So in other words, what we're showing here is, as you can tell, there's two tables. Top table is showing positive markers, the bottom is showing negative markers. What that means is these are markers that you would expect to see on this cell type. And so specifically what we're looking at here are mesenchymal stromal cells, or MSCs. And so there's three markers that they should all have, and then there's multiple markers that you're not expecting to see. And so what we did is let me just make sure I'm clear. These are kind of the recommended characterization markers by ISCT. And so the bottom line is all these cells were started from the same stock, but were expanded using different cell culture supplements. So that's what you see at the top. Obviously, FBS, fetal bovine serum is very common, AB serum, and then these are kind of different lysate products that are available. And so really what you see here is, based on the CD markers, regardless of what cell culture supplement you added, they really look the same. So looking at the characterization of them, you would say, all the same, or, all these cells are really the same. You can go ahead and go to the next slide. Now let's look specifically at potency. So we took the same cells, performed a potency assay on them. Specifically for these, we're looking at the ability to induce T regulatory or Treg cells from peripheral blood mononuclear cells, or PBMCs. And so the bottom line, or if you look just at the difference between uninduced in green and induced in dark blue, you can see there's a varying amount in the induction of Treg cells between the different cell culture supplements used. So this is using MSCs from 3 different donors. So we're taking that variability into account. But the bottom line is, you see different potency even though characterization is the same. So the bottom, so what you have is basically the cells look the same, but if you actually look at potency, you're going to have some differences in their ability. And in this case, the variation in potency is probably just due to the different cell culture supplement use. So this is why the FDA and others are harping on this. The fact is, by all your other assays, everything looks the same, but you could still have differences in potency, and that's obviously going to be very important to the patient, or the overall use of this particular therapeutic. Let me go ahead and go to the next slide. So what we want to talk about now is sort of some of the other strategies that we can kind of use to mitigate some of the risk. And one of these is to use qualitative versus quantitative assays, specifically for safety testing. So there is no current regulatory requirement that safety assays have to be quantitative. As shown in the graph on the left, you can see that we have the limit of detection, which is marked as LOD of an assay, which is a qualitative function, is always going to be lower than the limit of quantitation, or LOQ, as shown on the graph. The big question, though, is how much lower is it? Is it a minor difference? Is it a larger difference? It really kind of varies, but that's why we have the data there on the right. That table is showing where we determine the LOD and LOQ for several safety assays for different viruses listed there. And once again, this is looking or testing mesenchymal stromal cells, or MSC cell samples. And what you can see is it really does vary, but sometimes it's quite significant, where you have large differences between the LOD and LOQ, especially in regards to Epstein-Barr virus, where there's over close to a 30-fold difference between the two. So obviously, especially when you're doing safety and this type of thing, it makes sense that you're going to want the most sensitive assay you have to make sure that your therapy going into people is as safe as possible. So that's why it makes sense, when applicable, to really use these qualitative versus quantitative safety assays. So can we go ahead and go to the next slide? So kind of taking that concept a step further, we have shown that you can use automated, high throughput, qualitative infectious disease monitoring assays. And these are assays that are traditionally used for blood screening, but you can utilize these to test mesenchymal stem cells as an alternative to traditional quantitative assays. And so we basically ran the assays. We demonstrated that these automated, high throughput assays display acceptable performance in LOD, specificity, precision, and robustness. So the data showing basically these things, the LOD, specificity, precision, and robustness, can be found in our white paper. The white paper is titled Testing Strategies and Custom Assay Development for Cell and Cell-Based Therapies. And it's on the Facilitate website, but it's also available here on your console. It's listed as a handout, so you're welcome to take that and read that at your leisure. So the reason for these assays is that they generally produce faster results. They are traditionally less expensive to run because you're not running multiple replicates. You're not having to run all the quantitation standards. So less complicated and basically, less room for failure in that regard. And also they're automated. And so the advantage to the automation is you're removing as much of the human risk factor as you can. So it makes them so that, you basically have less human error in performance of these assays. So we naturally kind of looked at this approach because our background is really in donor screening and high throughput assays. So it made sense to us to utilize these assays to sort of remove, the possibility or issues associated with quantitation. And, most of the assays that are utilized, remember, these are coming from, developers like yourself or from academia. And so the bottom line is, those assays people have used in those industries have traditionally been diagnostic. Once again, because we're coming from a different approach That's why we thought about this, utilizing these assays. So that's really all I have at this time, and let me go ahead and turn it back over to Ward.
Thanks, Scott. So in summary, from our presentation today, making sure that you choose the right lab is really critical to the success of your development program. There is going to have to be some level of custom assay development, specifically making sure that we focus on a potency assay that gives, as Scott indicated, gives you an indication of the performance of your therapy, the effectiveness. We want to make sure, and we're encouraging people to look at a robust development process and making sure you define your specifications early as you are choosing a lab and manufacturing partner. These are really important factors in terms of getting your product to market as effectively and as quickly as possible. We have found that the qualitative safety assays that we use every day in the blood and plasma screening market are very effective for this type of testing. And they are significantly more sensitive than quantitative assays. There's no current regulatory requirement for quantitative assays, so why not use an assay that actually has better sensitivity and can be automated much more effectively? And then consider use of the high throughput qualitative assays, as we've indicated, in monitoring infectious diseases seems to make a lot of sense. And for us, as Scott indicated, we've been dealing with these high throughput assays for a number of years, and so a lot of familiarity with this. And so for us, it made a lot of sense to consider these types of assays as we were looking at supporting therapeutic developers. With that, I'm going to turn it back over to Georgie and ask her to open it up to the group for any questions.
Thank you for that presentation. I think that was a really great introduction to this discussion segment of today's webinar. Before we do get started though, I'd just like to remind the audience that you can submit your questions to our panelists via the Q&A widget on your control panel. So just to get started, you mentioned very early in your presentation about the importance of identifying partners that can help along the way. And I just wondered if you could speak a little more to the process of choosing a partner. So what would be your top tip or slice of advice to developers who might be exploring partnering opportunities? Apologies, Ward, I think we're having a few technical difficulties. Are you muted at your end?
Oh, I'm sorry. I was talking that whole time, and I guess I was on mute. Okay.
Apologies.
Sorry about that. It happened. Yeah, technology, isn't it? Wonderful. All right. So I don't know what I said, but I'll start from scratch. So back to your question, I think, what we've seen in our experience is for many of the clients that we've been working with up to this point, helping identify opportunity to take potency assays and move them into a more robust format, into a setting where those assays can be repeated over and over again. Because as you move your product through the clinical trial setting, and ultimately, I think the goal of every developer is to get their product to a commercial state, you're going to need to make sure that you're working with a lab partner that is able to really help you get to an assay that can be repeated and can be consistent and reproducible over time. And so automation is a big benefit. And so as we've been talking to a lot of our clients, we've also been talking to them about conversion of their potency assay. So being effective in doing that tech transfer and figuring out what's the best way to create an assay that can be repeatable, that's robust, that we know that we can effectively manage from a GMP perspective when you get to a commercial environment. So all of those factors, and I know I'm probably going a little outside the question that you posed, Georgie, but I think that those are really the key things, and hopefully that came through in our presentation today.
And just to stay on the same point, are there any key red flags that people should be looking for when having those conversations about partnering?
Yeah, I think one of the things that we've seen with a number of folks that have approached us is, does the laboratory you've chosen or you've targeted, do they have a broad understanding of the regional differences around the world? Therapeutic development isn't just focused on necessarily one market. You might want to launch your assay, or your therapy in the US, or you might want to launch it in the EU, or you might want to target Asia. And so depending on where you want to go, that Emmanuel mentioned this during his segment, that's a really important consideration very early in the process, because we can help, because of our experience, we do testing for clients that are globally based, and we're certified across multiple different regional regulatory agencies, not just the FDA. And knowing that and having experience in those testing markets, we can provide information that'll help therapeutic developers, really figure out and navigate the challenges associated with those environments.
Thank you.
Emmanuel, you want to add anything?
No, and I don't know if this is necessarily a red flag, but I would add to, as far as selecting a good lab partner, their ability to have a phase-appropriate approach and their understanding of the assay life cycle. That was one thing that I really tried to touch on in the first slide was, what does that assay life cycle look like? Being able to, really develop something that will hold up to that regulatory scrutiny that might be something completely different in the EU to Japan or Australia. And so, really just understanding that life cycle, the phase appropriate approach, it might be a little bit different for someone who's in a phase 1 or 2A versus someone who's in a 2B to 3 nearing commercialization. So being able to have that conversation and collaborate is important.
Fantastic, thank you. We have an audience question here that says that you mentioned regional specific differences and lack of harmonization regarding potency assays. So just staying with the idea of those regional differences, could you elaborate more on what those regional specific differences might be, and perhaps include some examples?
So I think someone who might have some experience of this is, so I have someone from our team, Joe Higdon, who is actually our Director of Quality for Advanced Therapies at BBG. And I'd like to see if he'd like to chime in on this question.
Yeah, hi, I guess greetings everyone. I get to kind of carry on Emmanuel's presentation slides from earlier. I think he did show a good example of how certain clean room expectations tend to vary across the different major regulatory bodies. I think you're also seeing a slight difference with difference of opinion with regards to data integrity aspects of different equipment, assays, things along those lines. And again, the ability to implement, justify, and defend various potency assays for a lot of these new therapeutic areas. It's, let's say, the onus is definitely falling onto the developer, the manufacturer, to not only prove that, like, say, the therapy is actually performing the way it is intended, but it's actually causing the benefit and not any of the other unnecessary side effects that we're looking for. This kind of goes past, like, say, the small molecule application, the biologics application, therapeutic proteins, and into much more complex areas. So that onus is definitely falling on the therapist, the developer, and the manufacturer to show that, yes, this is the expectation and the outcome that we're expecting or that we were planning on. Here's our proof of concept around that. Here's our method and our implementation strategies around those. And I think that is definitely the area that has the largest variability, just due to the the developing nature of the industry itself.
Yeah, to add to Joe's comments real quick, Georgie, if you don't mind, it speaks to the reason why it's so important to really understand what your laboratory's capabilities are. And why I say that is because potency is really a new area for a lot of people, and we've already done quite a bit of work internally to identify what's the best way for us to determine how a therapy is going to work, and how can we test for that? What are the methods that we can put in place? And I'm not saying that other laboratories haven't done it, but I think that looking for a laboratory that has experience in developing assays and doing so in a robust fashion so that the assay can be consistent and repeatable over time and is not tech dependent. A number of assays are performed now manually, and some of those assays can have a great deal of tech dependency. So some level of automation and making sure that the assay is repeatable, reproducible, is going to be very critical as you look for support with a potency assay that you might have come up with. The other thing to think about is the laboratory going to be willing to co-develop and collaborate with you to help you identify a viable and effective potency assay? And so that's another key thing to think about as you're identifying who you want to work with.
Thank you. I just want to move on to a slightly different track. So a key challenge that you highlighted, Scott, is that the testing demand is causing a capacity constraint. And I just wondered if you had any recommendations for developers in preparing ahead for a roadblock such as this, or perhaps when they should be implementing risk mitigation strategies.
That's a good question. Kind of like what I mentioned in my talk, and that is, I think, part of it is to be very proactive and obviously start thinking about assay development as soon as you can. And really finding that appropriate partner and working with them. A lot of it depends on sort of trying to figure out exactly what kind of assays they need, especially in the realm of potency. If there hasn't been, any work done on potency, or they do not have a well-defined assay, that's obviously something that you would have to work on right away. And that's probably going to be one of the assays. Usually, the key thing with potency assays too is, we're kind of used to the term when we say assay that you're only running one assay. So one of the important things to figure or think about with potency is that it's not out of the question. In fact, my bet is it will be very common that you're actually going to have several different assays that you're running for potency. So to your question, then it makes it very important to find a laboratory that has sort of the bandwidth to do that, and also focus on that early on, because usually that's a big hurdle. And then kind of what we mentioned a little bit before, some of it may also be the other kind of hurdle is that tech transfer. There are assays that need to be moved from the therapeutic developer to the testing laboratory. Does the laboratory have that ability? And kind of what we mentioned before is that even though the developer has those assays, developed and are performing them, more than likely, because you need to have that, make sure that they're consistent and reproducible and those things, you need to make sure too that usually that takes time, and usually there's some modification that has to be done during that process. So that's usually a kind of a common issue too that comes up, and you want to address right away, because those are kind of, anything about development, is going to take a little bit longer than things that you can just move over directly. So.
Excellent, thank you. And another element you discussed was actually that there will be some level of custom assay development in the area of potency. Are there any other areas where customized approaches might be key?
I think they're kind of important in all areas. It just sort of depends, and a lot of it just depends on exactly, the therapeutic developer's discussion with the regulatory agency, whether it's FDA or other. So what those requirements are and kind of how far down the road they are. A lot of the, like we mentioned and showed with the CD markers, a lot of the characterization assays, at least right now, are relatively well defined. So there's a general idea of what you're doing. Also in regards to safety, there's at least a general idea. And so those are probably less likely, but if there's any indication of that, that's obviously the best thing. The sooner you know about it, the better. But getting back to the potency thing, I do think that's probably the most critical in the development process. And really the reason for that is there's a lot of challenges around potency assay development. One being, like I mentioned, the concept that you probably or may have to do multiple assays in order to show potency. So, you got to make sure you have time and have the ability to develop what those are. But really, there's no universal one-size-fits-all potency assay. I don't know if that will ever exist because of the uniqueness of the products, which is a good thing, or the therapies. And so, yeah, and then the other thing is because of variability in the starting materials due to donor variability and cell line heterogeneity, that also makes it harder to show potency, but also part of the reason why you need to show potency. And then the last thing is there's really currently a lack of appropriate reference standards. So that obviously makes life a little bit harder too. So I think those are kind of the key things, but in all honesty, you could have, depending on, the uniqueness or what your therapy or product is, there may be, other custom assay development that you're going to need. So I think a lot of it is, just making sure that, you get with the right partner. And like I said, they have the bandwidth and are able to cover everything, because you may go into it, the collaboration thinking, oh, I only need help with potency. But more than likely, if you're tech transferring or doing anything, you're going to need quite a bit of help from that developer just outside, even outside of the help and development of potency assays.
Also, Georgie, Tony made a good point in the chat about the fact that developers, even if they've worked with a commercially available reagent, or they've adapted something for their purposes as they've been working on their therapy, those assays haven't been properly validated and set up to be used in a GMP environment. And so the developer, the therapeutic developer, may have been using good laboratory practices, but a GLP assay isn't going to meet the requirements from a regulatory perspective that a GMP validated assay will. And so, making sure that your laboratory partner has a lot of experience in implementing and consistently providing GMP certified assays is a really critical part of the decision process.
Yeah, very good point.
Yeah, thank you for making that extra point. I think also we did speak a little bit to some of the challenges in potency assay development were kind of covered in that answer there. So if we stick with the idea of customized approaches, I think the idea of customization can sometimes be associated with higher costs and expenditure in this field. So how can the cost or investment in custom assays here be mitigated later, perhaps in the process?
One of the things that we do as part of our process, if a developer approaches us with their therapy and they want the laboratory to work on their therapy, or they want us to move it into preclinical manufacturing, we take a really careful look at the assays that they have built, they're working with, and we determine fairly quickly if there is a way for us to do a tech transfer of that custom assay, and then is the assay going to give us the best performance and minimize variability. And if we can identify a commercially available, or we can adapt an assay that has higher levels of automation, we'll work with the developer, the therapeutic developer, to propose that we go through a process that we call test method suitability studies. And basically, it's a comparative study between the two test methods to verify, can we move testing to a platform that gives us that robust validation capable procedure. It's a really important step in the process. And again, it just speaks to starting the discussions with your laboratory partner early so that you can identify opportunities, because some custom assays that were built and set up by the therapeutic developer are not going to be sustainable over the long haul for whatever reason. And we try to ferret out those potential risks early in the process.
Yeah, the only thing I would kind of add to that is what we mentioned before, and that is we're also trying to actively find alternatives, such as, we talked about, utilizing blood screening assays as much automated and those type of assays that are possible. That allows us to be able to sort of cut down on costs. But Ward is, spot on in his comments. And yeah, a lot of it is, do you truly have to do customization? Is there a way to utilize something that's already currently out there, which we always try and do? And then, like we said, if there's not the, if you don't have the ability to do that, that's when we kind of move into that mode of doing customized assays. And then obviously through our method of test method suitability studies, trying to figure out how we can basically maximize that and make it as cost effective for the customer as possible.
And then I would just lastly add, as far as keeping costs down or making the process easier, phase appropriateness is a big thing, right? Because if you are phase appropriate in your approach, then all of the qualification and validation that's necessary in order to hold up to commercialization scrutiny, you can build in that more robust qualification later on in a phase 2B or 3, as opposed to if you're in a phase 1, that might not be required at that point. So having that discussion about phase appropriateness is also really good at mitigating your costs early on.
Yeah, absolutely. I couldn't agree with Emmanuel more. Phase appropriate testing is a really critical part of the process, and that will help keep the cost down. Because when you get to full-on GMP testing, it's when the costs, because for GMP, it's all about documentation. And so you've got to have all of the appropriate validation and documentation. And starting out with GMP validated assays right out of the blocks could be pretty expensive.
I think if we could just keep with the idea of phase appropriate validation here. I had a quick look at the white paper that you referenced in the talk, but also that we've provided as a handout for attendees. I just wanted to, yeah, as I say, stay with phase appropriate validation and just ask, like, what are the key considerations for developers, not just surrounding cost, but what are other things that developers need to be thinking about in that phase appropriate validation arena?
Manuel, want to give it a go?
Yeah, well, I mean, I think early on, you're not going to have that lot-to-lot comparison, right? So, from a robustness and repeatability standpoint, you're not going to have that data early on. So early on, you just want to make sure that the test and the analytical target profile of the test is defined, and that your assay can detect what it is that you're intending to detect. Over time, as you go down that the clinical trial phases, and as you are seeing that testing go from lot to lot, and you have that comparability, you're going to have more data to support that robustness part. So really early on, you're looking at it from a verification standpoint. Can the assay work as you've intended it to work as? But you're not going to have all of the data points necessary to fully qualify that assay as a validated assay. So that would be the simplest way that I could explain it. I don't know if Scott or Ward, if you want to say anything.
The only other thing I would kind of add is, and I agree with you 100%, I think just another way to kind of word it is that really during that early process development, you have a bunch of attributes that you're really trying to assess, and you're really trying to determine, hey, which ones are going to be the key ones or your CQAs. And then once you determine those, CQAs, that's really, where you get into more specific validation towards the later part. Because really in the beginning, you're kind of casting a wide net and just trying to see what are going to be, kind of the key indicators, the key assays that you're going to need. So kind of falls in the same thing Emmanuel said, just sort of a different way to word it.
So
Georgie,
oh yeah.
Yeah, real quick, before we wrap up, if there aren't any other questions, were there any additional questions?
No, we've come to the end of our Q&A. I just had a question for myself, really just wondering what you as a panel would like to see implemented to help overcome any current challenges in assay development, just to enable a slicker and easier development process in the future.
That's a great question. Scott, you want to start that?
I'll be glad.
Okay, well, I think, Georgie, it's that one, as much as possible where we can achieve harmonization and standardization in testing methods. I think that's going to be the key thing for a lot of therapeutic developers and for a lot of the organizations that provide the services for those developers, like laboratory testing and manufacturing. So as the standardization efforts progress to help the industry identify what are the right types of tests that can be offered consistently, whether it's the safety tests, which we have a lot of experience with, or the potency assays, which we also have a lot of experience with, that harmonization is really going to be key, I think. And I think the more consistently we can work towards assays and procedures, if you think about the clinical world, and what it used to take to run a glucose years and years ago. Now, hospital laboratories and reference laboratories around the world are standardized to a common gold standard for glucose testing. Just using that as kind of a simple way to break it down. This industry, the cell therapy industry, and people that are developing therapies using cell-based methodologies, we will get to standardized assay methods. It will take time, but it is something that I think is going to be important for the industry.
Yeah, I agree. That's obviously the number one thing that would help everybody. And then obviously, you know, and to sort of expand on Ward's use of the word harmonization, well, you know, obviously it would be better too from all the different regulatory groups that they basically harmonize together and all agree these are the key things. Obviously, that's a huge ask and will probably not happen, but at least having a defined idea over not just the assays, but platforms and really controls and really, you know, kind of the performance specifications, you know, even on your safety assays, you know, it must be able to detect, you know, this level of sensitivity. It's really kind of those type of things that will make life a lot easier. It's obviously a lot easier to customize and develop and build an assay if you know exactly what those specifications are, and if you know those specifications aren't going to change anytime soon. So that's really, you know, that's our, I would say, top of our wish list.
Yeah, I would agree. So Georgie, if I may, real quick.
Questions.
Okay, just wanted to acknowledge a number of members of our team, BioBridge and QualTex. Anand, Jennifer, Kamali, Zeke, Gavi, Chris, Tony, and Joe have all been contributors to this, as well as Sophie Barty, who is on the call as well. And just want to acknowledge their support and give them a quick shout out. So thank y'all. And Georgie, thank you so much for giving us this opportunity. And I hope that the participants today really found this helpful and appreciated the information that we provided.
Absolutely. And unfortunately, this is all that we have time for today. So I'd just like to apologise if we didn't get a chance to address any of our audience's questions. Before we do wrap up, I would like to take a quick second to thank Ward, Scott and Emmanuel for their insightful presentation and answers to our questions. As always, you can access this webinar on demand via the Facilitate Exchange website, along with many more of our previous webinars and related content. So until next time, thank you, everybody. And we look forward to seeing you next time.
Thanks, everybody.
Thank you.
Thank you.